that I actually put myself a reminder in my Outlook calendar to record the lecture. Because yeah, I've got a terrible memory, as you probably have witnessed. Okay, so what I wanted to do first is give everybody an opportunity to ask questions about linkage. So lecture, partly because of me screwing up the recording, but partly because it's a difficult subject. Um, not everybody may have gotten everything when they needed to last lecture. So I want to give people an opportunity to, actually, we could even do another practice exercise if you want for a 3-point cross. So if anybody has any questions about linkage, it's just math. Then we can spend, I don't know, like 15 minutes or so going over that first. Oh, well, that would be even more. Okay, so Alyssa is going to be doing the 3-point cross for us. Right, Alyssa? I think that counts as cruel and unusual punishment, to be honest, which is banned in like the Constitution or something. I'm not sure. It's all certainly not part of our pursuit of happiness. That's without a doubt. All right, so while you think about that, I'm gonna get busy drawing up a 3-point cross. Let's see what I can do. Mm-hmm. Now let's do D. Orbit dissertation. Actually, let's scrub that off. So, just thinking about stuff. I want to make this difficult, right? Because otherwise, hey, what's the benefit? Sorry for the wind noise. I've got a fan going to keep me cool. What's the other ones? All right. Okay, so and take a picture of this before I forget. So I'll do the same as I did last time and post these up to wherever I posted them last time. I can't remember. I'll find it. This is my, probably don't recognize these, some of the younger ones amongst us. It's a camera. All right, this is what we had before we had smartphones, or for the few of us like me who still don't have a smartphone. I actually got that camera when we were living in Australia, because my previous one, we went to a family birthday picnic, Oh, that's cool. And it was so hot, I think it reached like 43 Celsius. Don't know what that is in Fahrenheit, 120, something like that. It actually cooked the digital chip in the camera. So all the pictures ended up pink after that, which wasn't really a look that I was after. Okay, so here it is. Now remember, okay, let me quiz you first. What is the first step in Matt the professor's patented three-point cross? Hey, this is like being in one of those old, kind of Western prairies classroom where people on these tiny little desks and they chant back at you. Okay, find the parents. What's the second one? Oh, double recombinants. That's right. Because there are going to be a bunch of recombinants. We want the double recombinants. What does the double recombinants tell you along with the parents? Which one is moving? But there's going to be potentially, oops, check my number of fingers, three moving, right? Because we're going to have three different loci. Which one do you care about the most in terms of double recombinants? Almost. It's the one that is in the middle. So the double recombinants, what they tell you is essentially yeah. If the double recombinants are little a, big B, little d, the only way you can get that if these are the parents is if B is in the middle. So you need to know which the parents are because you need to know what the parental chromosomes look like, the originals essentially. And then the double recombinants tell you which order those genes are at in or on that chromosome. So the first two steps are about finding out gene order, and you cannot be sure that the gene order that you're given is the correct one because we may not know. I'm shrugging, you can't tell because I'm behind the board. Right? So that's really important. Remember, first two steps, all about gene order. Oh shit, dropped my board. Ah, crap. There you go. All right, what do you do next? What's step 3? Oh, that's right. I was going to say running around screaming is not a valid option. That's not going to help you in the exam. Right, calculate recombination frequency. But there's a wrinkle here. To calculate the recombination frequency between two loci, what numbers do you need to add together to find out the total number of recombinants for those two loci? Two lowest and, so the two lowest are the double recombinants. That's right, and the single recombinants for those two loci. Okay. Okay, I don't have anywhere to put my coffee now. My little cutie pie has come back after her fly hunt. She was licking her lips, so maybe she was successful. It'd be pretty neat if I had a cat that caught flies. Okay, so I'm going to hold up the top bit and then I'll move it to the bottom bit in a second once you've had time to take note of those. So we've got three loci, A, B, and D. Remember, the order in which you are given these loci is not necessarily the correct one. All right, let me know when you've had a chance to write those down. All right. Okay, so here are the ones at the bottom. There are no decimals there, just in case I wrote anything near one of the holes in the board. And because I'm not a sadistic teacher, I haven't jumbled up those, so they are in pairs. Oh, sorry. 72. Everybody got that? Let me know when you're done. All right. Okay, pipe up if you don't, and I'll bring it back up again. As I said, I'll post these board pictures to Blackboard, so you can go back over it just in case you didn't get it the first time around. Okay, so step one. What are the parental chromosomes? Awesome, because they have the highest number, right? So you'll expect an excess of parentals if linkage is occurring, right? And I probably wouldn't give you this data if we didn't think linkage was occurring. Okay. What are the double recombinants? Step two. All right. So, did I give you correct gene order? Nah. I wasn't trying to trick you. I'm just trying to illustrate that you can't assume it. Okay. So, what is the correct gene order? Peyton got that one right. The D has to be in the middle because it's the only, sorry, it's the only thing that's moved, right? So if you compare, if I write this up, where'd my board rubber go? Here we go. No, leave that alone. If you have A, B, D, and as a parent, yeah, you can see that, okay. Right, so if you have A, B, D as a parent, and A, B, small b, big D as a double recombinant, you can't get that if that gene order is correct. The only way you'd be able to get that gene order, sorry, you'd only be able to get that chromosome with this gene order if it's a single recombination between B and D. But you know that it's not. You know that it's a double recombinant because it has the smallest numbers. So the only way, oops, yeah, all right, calm down, cat. Is if these are the parents and So I need to move my laptop away a little bit further. There we go. And these are the double recombinants, right? So the genes are the alleles that you get is the same as what we wrote before, right? It's big A, big B, little d, small a, small b, big D. But the only way that you can get that, did I do this right? Oh, good point. Sorry. There you go. Fix that. The only way that you can get that D moving is if it's in the middle. And it's going to be the result of, let's see if I can see this well enough for myself, right? You have A, single crossover to B, and then another, or to D, sorry, and then another single crossover to B. So that's the only way you can get big A, little d, big B, which is that one. And because recombination produces the reciprocal, you're going to have little a, big D, little b, which is this one. Little a, big D, little b. Sorry, I'm reading everything backwards, which is hard when you have B's and D's around. Does that make sense? I'm trying to get it so the light doesn't reflect. Cool. And that's a really important point to get. So if it's hasn't kind of sunk in completely until now, that's fine. It's just important to make sure that you do get it. Okay, because we have to figure out gene order, because otherwise the numbers won't make any sense. Okay, so we've done gene order. Let's work out the recombination frequency. Okay, so let's do it for between A and D, right? So which ones are we going to be looking for here? Okay, so first up, we always have these in the mix, right? They're going to be added to both, right? Because they are result of recombinations between both loci. So if we want to do A and D, right, we're going to be looking for basically, where are we at? Big A, little d, little b, right? So we go big A, little b, little d. Those are the ones we want. So you have to add all those together and then express it over the total, whatever that is down here. All right, I'll let you get on and do that. See if I can hold this still. The 3-point crosses aren't trivial to get, so it's worth spending a bit of time, going through them. Repetition is the key. Well, all right, you got to show your working, Angela. Probably not going to be, there we go. What's the workings? That's okay. So you're probably working in proportion rather than percent. And recombination frequency is in percent. Map distance is in centimorgans, but they're essentially one and the same. Okay, so how did we get 16.9 centimorgans? And this is for the benefit of everybody else. Plus, I'll post this chat up to Blackboard afterwards too. All right, awesome. So let's work out the next one. All right, go show your workings, Peyton. Awesome. That is super cool. Come here. Okay, so let's draw this out on the board. I'll actually take another picture of that. So I think that'll be more useful. Okay. So we have, oh, that would be between, oh, actually, I'll let Payton finish that. Okay, so we have our gene order, which is A, D, B. Is it D, not P? It was, let's see, 16.9 centimorgans between A and D. And then 7.7 centimorgans between, there we go, D and B. And so one thing that you can see, you could actually do, hang on, no, I think that'll probably just confuse things. But essentially, including the double recombinants is the most accurate way for determining distance between points. So if you were to do this as a series of 2-point crosses, so you could do A and D, D and B, and then A and B, you wouldn't take into account those double recombinants. And so you get a undercounting, essentially, of the distance between A and B. Let's take a picture of that. Does anybody have any questions about three-point crosses? No? Cool. Okay. And what my cat is doing. So let's start on, and if you do after this, that's fine. Just send me an email, or we'll probably have time on Thursday to answer any questions, and hopefully, fingers crossed, depending on how well we do today, we may even have time for just an open question and answer session on stuff up till now, because Tuesday is exam 2 day. Right, so it'd be good to catch any issues before then. Okay. Let's see. Oh, shit, you don't want to see that. Shen. Okay. Let's move this over here. This is just me showing off, right? So we talk about doing recombination. This is unintentionally, because I just saved it before coming on here. This is an example of one of the crosses I did years and years and years ago. And so you can actually see, here's, oh yeah, that's that's how I did that. All right, this is actually a recombination cross on the same chromosome to get these two alleles together. This is one I want, this is a marker. And these two are very close together. So it's a very, very small number of progeny will actually have that chromosome. And I've got not hundreds of these, loads and 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 loads. And yeah, actually, this was one from a couple of summers ago. Did I get this right? Yeah, that one I never got it because it didn't work. So it doesn't always work, unfortunately. But anyway, that's just as a partly showing off as well, because it's kind of fun to talk about my work sometimes. I don't often get an opportunity to. But just to show that this principle of linkage and recombination is not an academic one. It is actually one that I've used quite a lot, actually, in my own research. And you can even do some real funky things, like some of those crosses were exclusion-based crosses, where I was looking to get two different chromosomes things together, but I couldn't see one of them because it didn't have a visible phenotype. And so you can look for the exclusion of another thing very close to that hidden, unseen allele. And basically, when you lose that, then you must have the other one, right? So you can actually do some pretty kind of funky genetics just knowing some of these principles, right? So it does have an actual application. Okay, so essentially, again, apologies for posting up the audio stuff late last night. I was away for the weekend and I had a bunch of other stuff that unfortunately took precedence yesterday, kind of morning and afternoon. So yeah, that's why they were late. But most of the beginning of this is kind of generally recap type stuff, and it doesn't hurt to go over these things again, come on, because they are useful. And so this is just a basic recap. We haven't talked about purines and pyrimidines yet, I don't think. Or if we did, I forgot. But just remember, pyrimidines are the small ones. So, and they have Ys in them. So thiamine and cytosine are pyrimidines because they have Ys, and they're single ring nitrogenous bases. And they're called nitrogenous bases because they have a whole bunch of nitrogens in them, basically. They're nitrogen carbon rings. And those nitrogens and the oxygens, those are really important in hydrogen bonding, which is what holds those two strands of DNA together. And also is the one of the mechanisms by which you get base specificity in DNA replication, why it's so accurate. Because you can only have, let's see, kind of jump around here, you can only have hydrogen bonds in this format between G's and C's. We'll talk a little bit about the numbers in a second, but just kind of riffing off that idea. And the same for A's and T's. So only these two, and they're actually going to be in opposite orientation. So technically the T or the A should be written upside down, right? Because they're anti-parallel. But you're only going to get those two hydrogen bonds if it's between those two bases in that orientation. And that's in large part the basic mechanism underlying DNA replication and how specific and accurate it is. There's lots of other cool stuff which we'll talk about. I think that's going to be PowerPoint 11, so maybe today or maybe on Thursday. But the basic underlying everything of DNA replication and actually transcription too, if you think about it, because it's the same principles, are these the structure of these bases and how they form hydrogen bonds, right, between this basically polar covalent bond with the oxygen. So this is going to be electronegative, and then the nitrogen here basically pulls those electrons away from the hydrogen. This is electropositive, right? So we get that hydrogen bond between these two. If you tried putting a thiamine next to this guanine, you can see, well, maybe this one, no, actually that wouldn't line up because it's an oxygen, right? We can't have a hydrogen bond between an oxygen and oxygen. And likewise here, we'd have it between a hydrogen and a hydrogen, right? Which also wouldn't work. So that's the very kind of nitty gritty detail as to why one base will slot in and base pair with another base, and only that other base, right? It's based on the size of the base, its orientation, and the hydrogen bonds that it can form. And that's really how DNA replication works, right? The DNA polymerase is not seeing what base is there, so it doesn't go, oh, look, there's an A, bring in a T, right? It's not a directed process. It's a, here's a space, this one comes in and it fits, clunk. Okay, that's good, make a bond. Move on to the next one. Okay, what fits here? Stunk, another one comes in and fits, right? If a different base comes in, most likely it's not going to fit. It's not going to be stable, so that phosphodiester bond cannot be formed, right? So this is why we talk about, or bang on about, depending on your perspective, these hydrogen bonds and how they're formed between the different nitrogenous bases. Because that is really, really what underpins, essentially all of molecular biology at the end of the day. And, oh yeah, another point too is that each bond has a certain strength, right? They're more or less the same, give or take. So if you have 3 bonds between G's and C's, it's going to take more energy to break those bonds, because there are more of them, than between A's and T's. And so if you want to pull apart 2 DNA strands, which bases would you expect to be more prevalent in that DNA sequence where you're going to be pulling it apart? Right, A's and T's. So, not A and T, because we don't want to go there, but A's and T's. And so you can actually look in like where transcription start sites are, which is actually a TARDA box, right, in bacteria. So it's a bunch of T's and A's. You can look at origins of replication, where DNA replication starts, and you'll find generally a higher percentage of A's and T's than you would G's and C's. Because it basically makes it a little bit easier to kind of pop that double strand open, so whatever needs to happen can happen. Okay, the other really important point to get, and this is another one worth reiterating, is directionality of DNA. And that's whether it's a single strand in this case, or a double strand in this case. DNA goes from 5' to 3'. RNA goes from 5' to 3'. And it's read from 5' to 3' too. So proteins follow the same order as well. And that is important for DNA replication, obviously. It's important for transcription, and it's important for translation too, because that's the direction in which the transcript is read. And in DNA replication, it's important because you can only add new bases to the 3' end of an existing nucleotide, whether it's RNA or DNA. You can't add them to the 5' end. So not just does DNA have directionality like that, but you can only extend it in that 3' direction. You can't go like so. You can't add it to the 5' ends, which causes issues later on because they're going in opposite directions, and we'll see about that in a bit. Anybody have any questions so far? Nope. I hope your hotel coffee is up to snuff, Angela. I'm starting to miss. No, yeah, I know. It's nothing quite the same as homemade coffee. It's like water. That's funny. Americans either make coffee that's so strong that you could use it to clean your car engine, or so weak you could see the bottom of it. I don't really know why they can't get it kind of in the middle. Oh, chai milk tea, that would be nice. Anyway, I've got a. Geez, it's only 11:36. I feel like there's a break coming up. Anyway, we've still got a bit more work to do. So a little bit about structure, DNA. So this is kind of like fine-grained structure. It's a right-hand helix, and a couple of things. It has a very even spacing, right? So each base is 0.34 nanometers apart, and it does a complete turn for, and notice it was like that because it's right-hand helix, for every 10. So every turn is 10 bases long, about 3.4 nanometers long. And this is an extraordinarily durable and stable structure. So much so that you can kind of go digging around in, ancient mud pits and find old stuff and be able to sequence it. That's how we've been able to, or one way at least, in which we've been able to kind of craft the evolutionary tree of the Homo genus, right? Because we're Homo sapiens, is by looking at that ancient DNA, right? It's how we, know that we have a certain percentage of Neanderthal DNA in our genomes, right? There must have been some kind of crossbreeding way, 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 way back in our past before we wiped them out like brutal savages that we are. So anyway, and the other kind of really neat thing to point out is that it's not an even twist. And so I don't know if I'm trying to remember if I have a picture of this. But I can, was it I think it's the trip. Is that a trip repressor? I kind of have to remember. It's a little while since I've thought about that. But at least one of the proteins that we're going to be talking about later in terms of bacterial gene regulation recognizes the like the kind of larger scale structure of DNA, which is this minor and major groove. Pardon me. And I can't remember for complete certainty, but I'm pretty sure that it binds into two major grooves, right? So it's not that DNA is a string of bases. I mean, that's how we write it because that's kind of the simplest visualization of it. But DNA actually has its own kind of topography, like, kind of the hill country. And certain proteins will recognize that shape as well as the sequence of that bit of DNA. And so that shape, that kind of alternating minor major groove is actually really important for a lot of DNA binding proteins. And there are a lot of DNA binding proteins. Okay, so this PowerPoint kind of bounces around a little bit, so I'm going to bounce around with it. And typically when we talk about molecular biology processes, I'm sorry, especially DNA replication and transcription, we talk about phases. And essentially you have to start the process. And in DNA replication, that involves its own series of processes, right? Same as in transcription. You have to kind of pop that DNA open and then start elongating it. And there's a whole bunch of stuff going on in these two. So you could kind of, I mean, it's not like distinct phases, they're a continuum essentially. And then you need to end it somehow, right? Which may be just when you get to the end, but also might be a directed process. A little bit of terminology. Replication fork is essentially where that parental double helix is being unwound into two single strands, and those single strands are being copied. And each replication fork or even replication bubble, because it can be in like a kind of a double arrow sense, right? Starts with a replication origin or origin replication that pops it open, and then that bubble expands, right, as the new DNA is being synthesized. So these are terms that we'll be coming back to a bunch kind of all the way through. Another point to emphasize, which, and again, a lot of this is recap from GenBio 1, but for many of you, it may have been a while, or, may have just forgotten because time has passed. But a lot of this stuff is kind of going over and refreshing your memory of stuff that you should have already learned at some point. And this image, let's move that over there, is a really neat one because it illustrates how, so this is essentially a replication fork, right? We have the parental double helix up at the top. That's split open. There's a bunch of enzymes that are involved in that, which we'll talk about in a moment. And then we have each parental strand is serving as a template for a new daughter strand. So essentially the new DNA, right, once this is done, is going to consist of this strand. So this is going to be one replicated chromosome or chromatid, depending on what organism we're looking at. And this is another one. So each new chromosome consists of a parent strand and a daughter strand, right? So it's semi-conservative. And this has a lot of implications too later on in terms of other stuff like epigenetic marks, right? Which are modifications to DNA that are inherited, right? And so, your parental strand will be the one that retains all of those marks, and you're hoping to copy them to the daughter strand. So we'll see how that's useful later on. We're also going to be talking about it's just a bunch of facts, right? It's kind of hard to get over that when we talk about a lot of this stuff. There's just going to be a lot of different enzymes that do different jobs. So what I find one of the best approaches is really think what's going on right now, right? What is going on in terms of this whole process of DNA replication? Are we starting it? Are we continuing it? Are we finishing it off? What's going on? So what needs to happen, right? We need certain enzymes at one point and we don't need them at the other. And so that helps, that kind of frame of context, I find, is a useful help in remembering what all these different things are and what they do. So for example, helicase is what unwinds that double helix so you get those two strands, right? DNA ligase is something that happens way later on that seals the nicks between the phosphate sugar backbone, right? So remembering those different things on their own is really tricky because it's just a bunch of facts. Remembering what they do and why they're required and where they fit in makes it a lot easier. So there's also, a bunch of words, and basically we have different ways of replicating depending on what kind of organism you are. So bacteria use what's called theta replication, right? So bacteria have circular chromosomes. A lot of viruses do too, not all of them, but some of them do. And also bacteria have other kind of extra chromosomal elements called plasmids. Quite often, antibiotic resistance is something that is stored on those plasmids. They're like an extra pamphlet where the chromosome is a book, let's say. So bacteria have this circular chromosome, and so they generally replicate it in what's called theta replication. And theta replication is called as such because this shape here, or even this one, looks like the theta symbol in Greek. That's pretty much it. And the reason why it looks like that is essentially you're going to start with one round and you're going to be copying two strands of that, right? And so essentially you go from that to kind of that to that, right? Where you end up with two replicated circular chromosomes. And then shortly after that, you'll get binary fission, and then you get 2 new bacteria. So every bacterial chromosome has a replication origin or origin of replication, often called an ORIC. I don't really know why. I don't know what the stand for. C stands for, rather. And essentially, bacteria can either do bidirectional replication or unidirectional replication. And that's really about, if you open up the DNA here and you just have one replication fork going around, it'll just go and it'll make your 2 chromosomes. Or maybe if you want to do it a bit quicker, you can have 2 replication forks which will go round and then they'll meet up at the bottom to generate those 2 chromosomes. Not really sure if bacteria can do both, a single bacterium can do both, or if it's something that's more delineated by species or type. There's also what's called rolling circle replication. And this is basically, looks a little complicated, but the idea here is you don't have to worry about the lagging strand, because what you're going to do is you're going to generate a whole bunch of kind of single-stranded bits of DNA, right? That's what these are. So you kind of cut the circle, flip that bit off, and then you continue replicating that strand using, say, the inner strand as a template. And as you get round, you get one linear piece that then recircularizes, which you see more towards the right on here, like here. And then that serves as a template for continuing that replication. So in theta replication, you have to wait until this whole process finishes to begin it again. So you start here, you end up, how did I do it before? Kind of with that theta shape, you end up with the 2, and then each one of these can do the same thing again, right? Theta shape, and then 2. And so you're limited by the speed at which you can finish this process, even with 2 replication forks. However, with rolling circle, you can kind of throw off lots of single-stranded templates, and then each one of those can then serve as template for replication. So this is a lot faster. And in terms of viruses, this makes sense, right? Because typically what viruses do is when they get into a susceptible cell, they want to replicate, as quickly as they can, burst or do whatever they do, or bud off, and then infect a whole bunch more cells. So their replication rate is an awful lot faster than bacteria or eukaryotic cells. And also they don't have to worry about such fine points as cytoplasm, right? Because they're not cells. They just have to get their DNA or RNA together and a whole bunch of proteins, and they're kind of good. So this is a lot faster than theta replication, and it's really restricted more to viruses and some plasmids. Has anybody got any questions so far? A little break. Yep. Okay. So DNA replication in eukaryotes, however, is a little different, both on a big scale and also at the actual details. The details we'll be looking at in the next PowerPoint. The big scale stuff, and also there's some issues with this too, right, that this causes. The big scale stuff is that eukaryotes have linear chromosomes. Some viruses do too, but we don't tend to worry about that so much. So this kind of circular kind of method of replication will not work. Also, just one replication origin is also not going to be very useful. So DNA replication in eukaryotes typically consists of lots of origins of replication, thousands potentially, because chromosomes can be Oh, they could be hundreds of thousands of bases long, right? And so you can't start at one end and go all the way to the other end because it would just take too long to copy your DNA, and therefore cell division would occur very, very slowly. And so what happens is that we have multiple origins of replication along a chromosome. Remember those sites which are a little easier to kind of pop open. And those replication bubbles will basically expand, from all of those origins of replication until they merge together. And that will form one larger replication bubble. And then those larger ones will merge together, and eventually you'll generate your newly synthesized, chromatids, your replicated chromosomes. Okay, so again, just to emphasize, nucleotides can only be added to the 3' region. So I'm going to draw this up. I'm not sure how well this is going to come out, but we'll see how we do. Ouch. This is certainly not something I'll miss when we go to face-to-face teaching again. Let's draw up. And we'll just assume that this goes to the, kind of continues on into part of the chromosome. We're not at the end. Okay, so this is our replication fork. We can fill in details and stuff as necessary if we need to. Top strand goes 5' to 3'. Bottom strand goes 3' to 5'. So just as a recap, what direction is DNA synthesized in? Oh, let me get rid of this for a second. 3'. Quit it. Go away. Come on. Sorry, I'm trying to get back to the PowerPoint. Yeah, there you go. Let's invigorize this a little bit. Okay, so one of the newly strands is going to be, not 3 to 5', 5 to 3'. It's going from this end over here to this end over here, right? And overall, the direction of replication fork is going to this way. Left should be, right? So what direction is the other strand going to get synthesized in? Is it going to go this way too? Or is it going to go that way? Don't forget, DNA replication is always 5' to 3' for the new DNA, right? It's always what you have to remember. We're talking about the new bit of DNA. So the other strand is going to have to go in the opposite direction because it's 5'. That's right. To 3'. it's going to go to the right-hand side, that direction, right? And that causes a pretty major issue because the whole deal is going this way. And these two strands must be synthesized at the same rate. So essentially, once this is continued on for a bit, let's make these a little bit shorter. Like so. This one is going to continue being synthesized. Right, if we imagine this replication fork moving to the left-hand side of the screen. But if we continue doing the same over here, we'd end up kind of going even further in the wrong direction. So we have to start again and produce another short fragment so that we don't lag too far behind, or we don't lag behind at all, this strand here. And so this one, my writing's going to be even worse, is called the leading strand because it's synthesized continuously. This is the lagging strand, not because it goes slower, all right? It's because there's more work than needed to finish off and make this into a complete continuous strand than there is for the leading strand. This is just for one replication fork, remember, or one replication bubble. Actually, no, just one replication fork, because presumably we're going to have the same going on the other side, right? And there are thousands of these per chromosome, right? So it just takes longer to finish this up. Can anybody remember what these fragments are called? They're short, like 200 to 300 base pairs, give or take. Yay! Almost. Okazaki. Osaki? No. Isn't that like a town in Japan? Or was that Osaka? Okazaki fragments. Where am I at? There we go. So these are Okazaki fragments. They're named Okazaki after the researcher that discovered them. I think I got it right. Okazaki? Yeah, I think that's right. And the basically the way in they were discovered is that there you go, Osaka, that's it. The way they were discovered is that if you radio label DNA, so you add radioactive nucleotides to the mix that are used to make the new strands, and you let DNA replication go for a little bit and then you stop it, right? You just pause it, or you stop any more the process from continuing. And you run this DNA on a gel to separate the different sizes, you'll get the parental DNA, which is huge, but you'll get one large band, right, which is the lead-in strand, and then you'll get lots and lots and lots of these smaller bands, right? So let me don't want to knock that over. Take a picture of this, and I'll draw out what I'm talking about. Ah, crap. Sorry, notice I just rubbed off something in the process. So essentially, if you, let's say, 0 seconds, right? You don't let any DNA replication occur. You'll end up having big band at the top, right? And remember, DNA is separated by size on gels, so the smallest band, smallest DNA fragments go furthest because they're able to make their way through quickest. So those are the parental chromosomes, essentially, right? If we stop it at, let's say 30 seconds, I can't remember the times exactly, but it doesn't really matter. You'll still have the parental, and now you'll have your leading strand, right, which is kind of reasonably big, one large band. And you'll have a lot of Okazaki fragments down here. Stop a little later. You know, obviously the leading strand gets a little bit bigger because it gets a little bit longer. And you still have these Okazaki fragments at the bottom, but now you start getting bigger and bigger fragments as those fragments get stuck together, right? And if you go long enough, I don't know how long it would be, let's say, go long enough, these lead-in and lag-in strands will be largely finished, and they'll be very similar in size to the parentals because they'll look exactly the same. And I don't know how much longer you get, and then essentially you'll end up with that, right? So you'll end up with DNA that looks just like the parents. And so these Okazaki fragments are a very temporary solution to the problem. They will get stuck together to make larger and larger fragments until they're all joined together, and you'll essentially get what looks exactly like what you originally started with, just twice as much. Does that make sense? And the guy, I think it was a guy, discovered this, or at least worked on it, was called Okazaki. The lagging one is the smaller ones. Oh, actually, good point. I'll make sure you can see it right. All of those are the lagging strand. So the lagging strand first gets synthesized, where's my finger, as Okazaki fragments, and it still does going along. And then, but as new fragments are being made, the old ones from here maybe, from 32nd, get stuck together to make larger and larger fragments until eventually you get one long continuous strand, which is essentially, you know, the finished complete product. Does that make sense? My cat's trying to tell me to play with her. She's got a toy in her mouth. I don't know if you can hear her. What? Quiet. Cool. Okay, where was I at? Okay, so let's just do a wee bit more, and then we'll break for a little bit, and then how much more we got left? Oh, let's finish this up, and then we can see where we get. Cat, shush. And God damn it, she's so distracting. and then we can see where we get in the second lecture for today. Sorry, I've got to go and sort this cat out because she's just distracting the crap out of me. Sorry. Never thought that the most challenging part of teaching online would be dealing with my cats or cat. Okay. Let's get that off. All right, so essentially you can see that deal here, right? And there's some good videos online which kind of show what the lagging strand is doing. But essentially it's kind of looped out and synthesized in a small fragment, finished, and then it kind of loops itself back in again, makes a small fragment, finished all the time that the leading strand is being synthesized along. And so when you look at this kind of in a bigger picture, let's look at it here, then you'll see essentially for each replication fork, right, we've got one fork going this way and we've got one fork going the opposite direction. You have leading and lagging strands for both of those, right? You have a leading strand up here on the top because it's going to the left, and the one on the bottom because it's going to the right. And the opposite strands are synthesized in those small chunks called Okazaki fragments, which also is the case in prokaryotes, right? You have the same deal. It's the basic same principles. You can't really get away from the kind of fundamentals of DNA structure, right? They're pretty much baked in. The only case where you don't see that as an issue, where there is no lagging strand, is rolling circle replication, because essentially the opposite strand is not synthesized until this outer strand is kind of cut off, recircularized, and then synthesized from that point onwards, right? If you kind of look back up here, for example, right, there is no synthesis of the second strand until you get over here and you've recircularized that external strand, right, which was originally the bit on the outside on this structure. And then you can use that as a template to make the other one. So for rolling circle replication, there is no lagging strand, right? It's a lot simpler, faster too. And it gets around the whole lagging strand issue by just essentially synthesizing one strand at a time. Because prokaryotic and eukaryotic DNA is so large, especially eukaryotic DNA, you can't do that, right? You have to do both strands at the same time, right? You have to finish both strands at the same time too. And I think that's a pretty good point to stop at. Does anybody have any questions? Oh, awesome. I'm going to go strangle my cat for a little bit. He stops being such an annoying little furball. And let's meet back in 15 minutes. So 12:25. Oh, God damn it, Noel. Awesome. I'll go see what my cat wants. All right, see you in a little bit. All right, where are we at? Let's get. All right. So this one is the chunky version, essentially. So what we looked at last PowerPoint was really the overview. Like, what are the general principles going on, the general issues, things like that. Oh, you took the dog for a walk? That's neat. I scratched my dog's ear. There you go. That was pretty much all I did, and wrote a text message. It takes about 15 minutes on my phone, by and large. Anyway, so what we're going to do is have a look at DNA replication in prokaryotes first. Part of the reason is that they're a little bit simpler to think about what's going on, and also there's fewer things involved too, so it just makes it a little bit easier. Sorry, I just saw Alyssa. If that's what you call stressful, that's super awesome. Sorry, that was a little bit harsh. Forget me. Just the guy on the screen. Anyway, having to write a text message, go for a pee, and look after the dog, and eat a biscuit, cookie, and get some fresh water. Now that's a lot of stuff to do in 15 minutes. Anyway, so essentially we've got the same kind of deal as before. We've got initiation, unwinding and elongation, and then termination, right? And so again, remember, these are a continuum of processes. They don't happen in like, discrete time periods, but they're just like one thing leading into the other, just helpful to break it up. And so essentially in E. coli, the point here is that we're going to be starting this process at an origin of replication. And the way in which we do that is we get a bunch of initiator proteins. I don't know, they presumably have a fancier name than that. And we're going to wrap the DNA around those proteins. And what that does, essentially, it's like kind of inducing a coil in a rope, right? And so you're making it more coiled. And in DNA, at least, what that does is it actually produces the opposite coil somewhere else. Pardon me. And that opposite coil occurs in this region of DNA kind of just next to the origin of replication, which has a much higher AT percentage than elsewhere. Whatever the overall AT percentage or GC percentage is, there are more A's and T's in that region. And so essentially what you're doing is you have some DNA that's kind of coiled like this. You induce a coil over here, which then pops it open like so, right? So you've gone from being coiled DNA, you've kind of twisted it essentially. And so you now twist it so that now you have a small stretch of unwound DNA, right? Where each strand isn't directly kind of base paired with its partner, the opposite. And so what that does is it allows other proteins, and there's two kind of groups here, to recognize and bind that single strand of DNA, because you don't find single strand of DNA elsewhere at any other point other than in transcription, and that's kind of masked by RNA polymerase, essentially. And so as soon as that DNA is unwound and it becomes single-stranded, then you get the unimaginatively named single-stranded binding proteins come in and they hold that open, right? Because essentially what DNA is going to want to do is it's going to want to snap back and be a helix again, right? Those hydrogen bonds will want to be reformed. And so to keep it in that position, we're going to have to stuff those strands with proteins which stop that happening, right? Which physically keep the DNA apart. And that's the job of the single-stranded binding proteins, but we also have other proteins, and these are really important ones, called helicases. And the role of helicases is to continue that unwinding process, right? They are what essentially drive the replication fork outwards, right? And so you'll find one at the base of each fork. You can see there's one here and there's one here. We still have single-stranded binding proteins. You know, they are present too, because again, we don't want any part of that bubble to snap back. I'm feeling all guilty for ragging on Alyssa.
That's okay.
I guess I probably shouldn't say I'm used to doing that with my kids because that makes me look even worse. Anyway,
I was laughing. It's just
yeah,
obviously I was going to keep it on mute. So
it's all in with good intent. So those single-stranded proteins, they're binding proteins, they're basically temporary until they're forced off the DNA, the single DNA strand by DNA polymerase. All right, so they're not a permanent solution. They're just there until the new strands can be synthesized, in which case it's no longer an issue. You don't have to worry about it. Now I want to introduce, or perhaps reintroduce, if this is fresh in your memory, another protein. This is called DNA gyrase, which is a type of protein called a topoisomerase, which is a pretty fancy word. And so essentially, and I wish I had a piece of rope here. Yeah, I don't think I've got any that would be useful at home. Essentially, what the problem is if you unwind the DNA, you kind of pip it open from that, right? You've done that by coiling it more somewhere else. But as soon as you stop doing that and you start kind of doing more and more unwinding, right? So you start extending that replication bubble. What you're going to do is just upstream of that fork, that DNA is going to start getting all twisted up, right? It's going to start coiling on itself. Because if you take coils out of one part, they don't disappear, you push them somewhere else. And it's very much the analogy I like is if you get a bit of hemp rope that has 2 or 3 strands, you stick a screwdriver or pen in it and tie it to one end and you kind of push that pen along, right, to open up those strands, then you'll see the other part of the rope, it will start spiraling around itself until it kind of ends up basically like a small scrunched up mess. And so DNA gyrase does a really important job and it relieves supercoiling, right? So lots of coils equals supercoiling. And supercoiling is actually used particularly by bacteria, I think, to reduce the space of DNA, but generally speaking, you don't want it. And so what the DNA gyrase does, essentially, if you can imagine, right, we're getting all twisted up, right? So we get that supercoiling. What DNA gyrase does is break that DNA, either one strand or both strands, holds it together so it doesn't fall apart, and then just lets that supercoiling relax. Right, I actually did it earlier. My headset cable was getting all coiled up, so I just took it off my head, and you just hold it up like so, basically just let it unwind so that it's not a coiled mess. And that's what DNA gyrase does. And then as soon as that rotation has occurred, right, and those extra coils are now removed, right, because they've dissipated, then it sticks those the DNA strands back together again, right? And so it does that all the way along, just in front of helicase, which is separating those strands. So helicase is like the screwdriver going along, and essentially DNA gyrase is constantly cutting the rope, sticking it back together, cutting it, sticking it back together, as those coils kind of get pushed further down the rope, right? So it's a pretty neat process, to be honest. Okay, any questions? Questions? I'm going to take this at a nice pace because we've got time on Thursday to finish it off, and I don't want to go too fast. So I'm kind of timing myself so that we get probably about halfway through this PowerPoint today because there's a lot going on, and maybe get DNA repair done today, maybe not. If not, we'll do it on Thursday with homologous recombination. Okay, so one of the real big issues, well, there are lots of different issues to overcome, right? Which is why this process is so complex. One of the big issues, come on, is that the main issue is that you need a 3' OH, right? If we go back to, where are we at? Over here, right? We need this. Actually, even once you put a new nucleotide on, you have another 3' OH, the one that was originally at the bottom of the new nucleotide. So you need that 3' OH, otherwise you cannot form a phosphodiester bond. Not possible. The problem is that, at least in bacteria, DNA polymerase cannot just add a base to a template strand. There has to be something already there to provide that 3' OH. Otherwise, that base just falls off again. And so the way in which that is overcome in bacteria, eukaryotes have a slightly fancier way of doing this, is by synthesizing an RNA primer onto that DNA. So we have a DNA-RNA hybrid, right? We have our parental strand down here, our template, and we have our little stretch, like 20, 22 base pairs or so of RNA, right? That's synthesized by another enzyme called primase, right? Which is an RNA polymerase. It's not shown here, but essentially, if you imagine that this piece here is RNA, right, synthesized by the primase, which is typically hangs out with the helicase right at the beginning of the replication fork. If this is RNA and this is DNA, right, so now we have our 3' OH provided by the RNA, and DNA polymerase comes in, and now it adds a DNA nucleotide to it, and then DNA polymerase just keeps on doing so, right, until it runs out of template DNA to copy. And so that RNA primer is essential for DNA polymerase to do its job in prokaryotes. Also causes a lot of problems later, which we'll get to, because we don't have RNA in our genome. We have to get rid of it, right? So that causes other issues. So essentially, that has to happen for every bit of DNA that you want to synthesize. So for the leading strand, right, which you can see here at the bottom going right and at the top going left, that primer only needs to be synthesized once, right at the very beginning as elongation is started, right? For the lagging strand, it has to be done every single time you release the ligand strand, and then you restart the synthesis of the ligand strand, making those small Okazaki fragments. And to do so, that primase is at the very beginning at that replication fork in a complex with helicase. So there are a lot of complexes involved in pretty much any molecular process. It's not just one enzyme on its own doing something, typically, although DNA gyrase is kind of an exception. Typically, you get lots of different proteins kind of stuck together, each with a different role. Helicase for opening up the DNA, primase is sitting there with it, and as soon as it's opened up, it makes a new primer, and then DNA polymerase comes in and then runs with that to make the ligand strand. So it's a very complex kind of process essentially together. Let's see. Just give me a second. I'll see if I can find WeHi. Let's see. Okay, I'm just looking up a cool video. Where are you at? Here. It's over here. Oh, that's something new. So this is from a place in Australia, actually in Melbourne, called the Walter and Elizabeth Hall Institute, I think, with WeHi. Okay, so let's. Oh, can I share that? Share. Okay, can you all see my browser? Yeah, hopefully you're not going to be too traumatized by all the tabs. Read some NPR. We've got some other stuff here. Anyway, that's not important. BBC News. So what I'm going to show you is a little animation, right? So this is a molecular animation, so it's supposed to be as close as possible to how we understand it works in reality, right? But as an animation, so we can visualize it. And this is typically the one I use whenever I come up to this subject. Shouldn't worry about the noise if you can hear it, because it's just like stuff. Okay, so essentially to give you a bit of framework, we have the DNA coming in from the left, and the newly synthesized DNA is getting kind of spat out top and bottom, essentially. And what you can see here is there will be an enzyme complex at the beginning. I'm not sure if it shows DNA gyrase, which will be the helicase and primase. And then there's a whole nother load of proteins, including the top and bottom, like a kind of barbell, I guess, in a better way of putting it. That's the DNA polymerase, and they're tied together so that this synthesis of both strands is linked, right? We don't synthesize one strand and then another strand separately. They're both synthesized together, right? And so at the bottom, you'll see the leading strand, which is being synthesized continuously. Ligand strand is different. I'm just going to let that play for a little bit so you can watch it for a little bit, and then I'll go back through it. This is such a cool little animation. I love animations generally. So, essentially what is happening is you have the helicase and primase. The DNA is coming through that, gets split into your two different strands, right? One strand is being synthesized continuously. That's your leading strand. The other one cannot be synthesized continuously because you want to synthesize it in the wrong direction. So what happens is it loops it back kind of round, like so. So even though it's going in that direction, it's going to be synthesized in this direction for a little patch. And what you can see is this is the bit being synthesized here, right? But it's going in that direction. It's going towards the left. And you can see just at the top of that frame a previously synthesized Okazaki fragment, right? Because essentially it's doing it backwards, right? And it will walk into what it has already done. And so once that DNA polymerase hits the beginning of the previous Okazaki fragment, the whole loop is flipped out and then brought back in with a new primer and synthesize that fragment until it hits the beginning of this fragment, which we're just finishing up, right? So just watch that for a second. So it gets kicked out, and then it's brought back in, synthesized, hits the previous one, gets kicked out again. And so you can see that going in. As soon as we're going to get the old one, gets kicked out, gets brought in. So really what you want to be doing is looking at what's going on the left. What's on the right is completely uninteresting because it's just one continuous synthesis process. The difficult part to get is the bit on the left where we're making those Okazaki fragments. Oh, I think this is some new stuff. What's this? Yeah, I'm not 100% sure what the different bits are doing. I think the light green is DNA ligase hanging out there until it's needed. So if we go back a little bit, let's see. So if you look at that green bit, it stays attached. Crap, this is so fast. And see just there, that green, oh, okay, give me just a second. This green part, I think, don't know for sure, but I think that's DNA ligase, which we'll get to in a little bit. This blue, light blue part on the left, that's the helicase. The darker blue purplish part, I think, is the DNA polymerase. So I think this is going to go in a little bit. Right, so again, this is the bit that's spinning is the helicase because it's kind of rotating as it's kind of forcing those two strands apart. I'm pretty sure, just as an educated guess, that this green part is DNA ligase, which is hanging around, and you'll see that it's actually left behind. It may also be another type of DNA polymerase 2. I don't know whether this is eukaryotic or prokaryotic. That's a kind of a wrinkle because it depends essentially because there are different mechanisms. But something that's involved with finishing off the process and getting rid of that RNA primer. And this purplish part is the DNA polymerase. In here must be a primase of some sort if it's prokaryotic, but if it's eukaryotic, then it's a different DNA polymerase. So again, don't worry too much about all these individual things, other than the helicase, which is neat, and the fact that you have two complexes which are synthesized in the DNA that are held together, right? They're joined together by this very pale blue bridge, essentially, right? And that's what makes the whole thing go together, right? So both strands are synthesized at the same time. What this video is really, really useful for is showing how the lagging strand is synthesized, right? And that's arguably the hardest thing to understand about this whole process. The lagging strand has to be turned backwards on itself. So essentially, it's pointing in the same direction, right? So if you have, let's do it this way, right? This is the leading strand. I can't bend my arms like that. Like that. The lagging strand is bent back on itself so that it's being synthesized in the same direction for a little bit as the leading strand. But you can't do that forever because essentially you run into what you've already done. And this animation is a really neat way of showing that. Because you can just see, hey, come back. Hang on, someone once told me. Ooh, yay, I remember this. Someone told me this in class once. I've got a terrible memory. Let's slow it down. Okay, so oh, shit, I'm pointing at the screen. That's ridiculous. This is essentially the 5' end of the previous Okazaki fragment that was synthesized, kind of a little bit way back, right? So we're kind of moving to the bottom left in the screen here. So this would be something from when it was in the top right. And what you'll see is that as that DNA polymerase synthesizes this new Okazaki fragment, which is this one just here at the kind of the top center from these 3 strands, it's going to run into the beginning of the one it's already done. And as soon as that happens, the loop is released, and then we're going to grab another bit kind of around here. We're going to make a new primer, right, so that we can start the process again. And then it's going to get looped back into the DNA polymerase, and the whole process begins again, over and over and over again for the lagging strand. So just watch this, and I've slowed it down, technology, so you can see it a little bit more clearly. Gets kicked out. Shit bits. I'm going to slow this down even more because I can't use my mouse pad quickly enough. Whoa. See, gets kicked out. All right, this is a little bit too slow. It's going to take forever. Okay, so now we're going to hit the previously synthesized part. And I think you can just see a primer being made here. I'm not 100% sure. Yeah. And so that grabs it, starts again, and now you can see that new strand being synthesized. And in a second, you'll see that one that it just finished here, kicks off, starts the whole process all over again, over and over and over again. Isn't that cool? It's so much easier when it's slowed down. I'm glad I remember that. I just want to see what's over here. What's this? This is a bit of the video that I haven't seen before. Oh, I think this is transcription. Yeah. That's pretty cool. I remember. I'll bookmark this so we can watch it. Ooh, that's cool. I'm not entirely down with the music though. I don't know why they need to put noise to this. But anyway, does anybody have any questions about the whole lagging strand deal? Yeah, I like it a lot. It's a super hard thing to try and explain in words. That's the YouTube video, and I'll post these chats up to Blackboard after everything's all finished, so that you can have a copy of this too. Okay, let's get back to. Oh yeah, that's another And there's also one for photosynthesis too, which is really, really helpful to kind of put all this stuff into context, right? Because otherwise it's just a collection of facts which don't necessarily make sense. Right. Oh, cool. I'll have to have a look at that one later. Might be useful for bookmarking it for GenBio 1. So you already know this because I told you already. But in prokaryotes, at least, there's 5 different DNA polymerases in E. coli. I'm not 100% sure about all prokaryotes, but at least these ones are present. And so basically they have different roles. And that's the same for eukaryotes too, but it's just a little bit more complex. So DNA polymerase 3 is the one that, well, actually, let's go back here. This is DNA polymerase 3. This is the one that synthesizes the bulk of DNA in prokaryotic DNA replication. We have another one, DNA polymerase 1, which is the enzyme that removes RNA primers and fills in the gaps, which we'll get to in a second. And we have a bunch of others that are involved in DNA repair, mismatch repair, stuff like that. So essentially, to finish this up, we have to get rid of these RNA primers, right? So if you imagine this, right? So this is a completed ligand strand. We've got a new one being made here. Once that is all done, you're going to have 2 issues. We're going to have RNA primers scattered all over the place, which is not good because we're, double-stranded DNA, not DNA-RNA hybrids. And also, these fragments are not joined together. So if you were to leave it as is, right? So that's like back in the 30 seconds part of that Okazaki picture that I drew, and you were to start DNA replication all over again, you would not have a single piece of DNA to copy. You would have lots of little bits to copy. Same in transcription, actually. And so it'd be like kind of laying a whole bunch of railway rails and then not bothering to join them up with each other, right? That'd be disastrous, if you're a train driver at least. And so what we have to do is fix those 2 problems. We need to remove the RNA primer, and we need to seal the gap in the backbone that is between those different fragments. So that's what DNA polymerase 1 does. What it does is it comes in, so it recognizes those DNA-RNA hybrids, and it digests or removes by hydrolysis those RNA nucleotides. And so it takes out an RNA nucleotide, puts in a DNA nucleotide, takes out another RNA, puts in a DNA, until it fills that whole gap which was taken up by the RNA primer. So that fixes one problem, getting rid of the RNA part of DNA replication. The next problem is that those 2 DNA fragments, so these are each Okazaki fragments here at the top, are not joined together. So if you were to denature that DNA, by heating it to high enough temperature to break those hydrogen bonds between the strands and just separate them, you'd have one continuous strand and lots of separate chunks, those fragments. And so you need to stick those fragments together. And that's the role of DNA ligase. And so DNA ligase uses ATP, so molecular energy essentially, to create that phosphodiester bond between the 5' end, or the 5' phosphate essentially, of the at the start of one Okazaki fragment, and the 3' end, 3' OH of that newly synthesized little filler part. And DNA ligase literally just joins them together. So it's a continuous series of phosphate base, phosphate base, phosphate base. It's not phosphate sugar, space, phosphate sugar, phosphate sugar, phosphate sugar. Now it's just a continuous series of phosphate sugars, phosphate sugars, phosphate sugars all the way along. And once that is done, more or less, DNA replication is finished. There is actually an extra little wrinkle that can, in some bacteria, actually forces the end of that DNA replication. Where's... Oh, it's a little bit further down here. Okay. Anybody have any questions so far about any of that stuff? Just take a little breather. So now we have, where do we take it? Let's take this to, let's say around 1:30, I think is good, because otherwise people are going to start like flaking out because there's a lot of stuff to take in, and particularly trying to do it in like one solid chunk is not trivial at all. I'm sorry, catching up on my my sleep. Well, not now, obviously. I'm awake now. Be pretty impressive actually to do this while I was asleep, like sleep teaching instead of sleepwalking. Okay, so what's next? All right, at the very least, what I want to do today is talk. Even while making mac and cheese. The little one. Oh, can you make some mac and cheese for me? I'm really hungry. Stick it in the post or something. I live by my stomach. For some people, eat to live, and others live to eat, and I'm definitely the second one. Okay, so a couple of things to finish up. One is accuracy, right? And the other one, maybe we get to it today, maybe we'll do it on Thursday. Hopefully, we'll get to it today. Is the differences in eukaryotes, sorry. Because this is all about prokaryotes right now. Tell my brain is starting to fade. Okay, so essentially one of the big deals in DNA replication is we need it to be accurate because this has to happen many, 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 many times. And even though in an evolutionary sense, mutations are pretty handy, right? Because they are what natural selection and other things can act upon. In terms of an individual, mutations really suck. You know, mutations are generally speaking not good for you. If they are going to have any effect, there's generally a bad one. And so you don't want to have to go through thousands of rounds of DNA replication and cell division to end up as a adult organism or functional organism and accumulate thousands and thousands and thousands of mutations along the way, right? You really want to have the same set of instructions at the end as you do at the beginning. And so DNA replication has a lot of mechanisms to fix this. RNA replication does not. So if you think about influenza, actually as a great example, is an RNA virus. And so every time influenza genome in terms of RNA is copied, it accumulates a bunch of mutations. HIV is actually the same too. That's also a RNA virus. And so that's why vaccines against influenza and HIV, we still don't have a vaccine for HIV, are so difficult to make because essentially every time that virus replicates and then gets into another person and replicates, it changes. Some of those changes result in the virion not being functional, which is like yay for us. but mostly it just changes the some aspect of the virus, so our immune system does not recognize it. That's why every year you have to get a flu shot, because not that the vaccine doesn't work anymore, right? That, the response in you, but it's because the virus is constantly changing, because it's constantly mutating and showing different forms. So your immune system doesn't recognize it essentially, and it has to start the whole uh, adaptive immunity process from scratch. So every year, scientists make their best guess at which is going to be the predominant mutant form of influenza, and they make their vaccine against that. And sometimes they don't get it right, which is why the vaccine varies in how effective it is. So RNA replication is incredibly error prone, which is not necessarily a bad thing for the virus. But in DNA replication is very accurate. And so inherently, the whole base pairing aspect of DNA replication is a big part of that, right? So it's just very difficult for the wrong base to be incorporated. But we also have a bunch of other stuff going on. So generally speaking, the error rate is 1 in every 1,000 to 1 in every, what's that, 10,000? 100,000 maybe, depends on the species. It varies. So between 1 10 to the 4 and 10 to the 5 typically. And that's just the inherent accuracy of incorporating bases. The overall error rate, particularly in eukaryotes, is about 1 every billion to every 10 billion bases. And so part of the difference is essentially DNA polymerases are able to correct their own mistakes. And that's called proofreading. It's like basically you go back over your writing as you write it and you correct errors as you make them, like backspace. And so essentially that's what these enzymes have. They have a backspace function. And so this is our newly synthesized strand here on the right, as that goes along, if somehow by chance the wrong base gets incorporated, it will look weird, right? It won't fit. 16. I don't actually know what the overall error rate for prokaryotes is. So I know that it's about 1 every billion to 10 billion in humans, for sure. I'm not 100% sure what it is in prokaryotes. It's a range anyway. Some DNA polymerases from prokaryotes have like 1 in every 1,000 errors. Others can be as good as 1 in every 100,000. It just depends on the particular species, essentially. So yeah, there's a fair degree of kind of hand waving and whiffle waffling. But the main point from this is that you have your base accuracy, like this inherent in DNA replication. You improve that with proofreading, so it fixes the errors as it goes along. You improve it further with mismatch repair, which we'll talk about a little bit. And all of those added onto each other gives you your overall rate of accuracy or error rate in DNA replication. And I know that in humans, it's about three errors every round of DNA replication, give or take. And we have a total DNA content every cell of about 6.5 billion bases. So that kind of fits within that general kind of general range, I guess. Does that help? Cool. Yeah, it's funny because it's like I've got these So from just working with doing PCR, different DNA polymerases for PCR, which come from bacteria, um, actually hyperthermophile bacteria that live in hot springs, have varying rates of um, accuracy. And it's actually a trade-off between speed and accuracy. So the faster an enzyme is, the more likely it is to make mistakes, and vice versa. So there are some which do a really, really accurate job, but they take forever to do. So yeah, that's kind of the context in which I know that stuff is really just from like looking through spec sheets on DNA polymerases for PCR. So anyway, that kind of base accuracy of just base pairing can be improved further by proofreading. And so essentially a DNA polymerase has part of the protein. I always think of it like on its butt. I don't know why. It doesn't have to be. But essentially what happens is that once an incorrect base is incorporated, it kind of has a little bit of a like speed bump. And it goes, hang on, something's not right. And it backs up a little bit and it cuts the phosphodiester bond through hydrolysis, right? So it actually cleaves off that speed bump, that incorrect nucleotide, kicks it out, and then starts again. So essentially it's kind of dung, 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 chugs along and goes huduk over something. Then it backs up. I don't know if it has a beeping sound like they do on like dump trucks. Beep, 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 beep. Cuts it off, lets a new one come in, which hopefully is a correct one this time, and then starts the process all over again. That's what proofreading is. Very much the case, you tappity, tappity, tappity down here is like, oh, crap sticks, I've put the wrong thing in, backspace, backspace, backspace. And then you start writing again, hopefully correctly this time. That's all it is, essentially. And it's a distinct part of that protein structure. Does that make sense? Awesome. Okay, and so the final part, right, to get up to that total overall accuracy of 1 in a billion to 10 billion, give or take, is mismatch repair. Okay, so do you remember when I told you about DNA methylation, right? The difference between parent and daughter strands. So essentially, every DNA strand, yours included, right, in your cells, has little marks. We'll talk about these when we get to epigenetics later on. And those marks are not present in the newly synthesized strand, the daughter strand, until a little while later, once they've been copied over. And so essentially that difference is what's used to recognize an incorrectly added base. And so sometimes this, this speed bump kind of deal doesn't work. And so, perhaps it's towards the end of a shift for that DNA polymerase driver or whatever. I don't know. They're just like, oh yeah, whatever, don't care about that. And kind of continue on the process. And so that incorrect DNA nucleotide is incorporated and the whole process continues on. And so that's called a mismatch. And typically what that generates, as you can see at the top, is an incorrect kind of shape to the double helix. It doesn't fit right. And there are enzymes that are called MUTs or mutes that recognize two things. One, that mismatch, that incorrect shape. And the second one is they are able to distinguish between the correct strand, which is the template or the parental, and the incorrect strand, which is the daughter. You imagine if this continues going on for long enough and those methylation marks are added, you can't tell between the two, right? It's like 50-50 which one is the correct one. But early enough after DNA replication, those marks have not been added. And so you can say, this one with all those old like chicken scratchings on it, that's the parental strand. This fresh and shiny one down here, that's the daughter strand. So that's the one that we have to fix. And essentially what they do is they make a cut in the daughter strand, and they, like a Pac-Man, kind of goes along eating up DNA bases and just like kicking them off until it gets to the incorrect one, right? And it removes that one too. Then another DNA polymerase, one of the many earlier that we talked about, comes in and fills in that gap. DNA ligase reseals it, and that problem is fixed, right? So that's the last kind of part of this DNA accuracy deal. Did that make sense? Again, there's two parts: the shape being wrong and being able to distinguish between old DNA, the parental DNA, and fresh DNA, which is the one which has the error in it. All right. Okay, not much more left. And this is essentially a summary of that. Okay. And also, again, it's worth pointing out that if mismatch repair doesn't catch this for whatever reason, then that error will become incorporated into the genome. So that then is a permanent mutation. So when that cell replicates again, or that cell divides again and the DNA is replicated, those two strands, the parent with the original base and the daughter with the new one, those will get copied. They're now both the parents, and that change in that daughter strand, that will then get copied into its daughter strand or granddaughter strand, I guess. And that mutation is now part of that genome, at least for that particular branch. The other one with the original parent is going to remain unchanged. Okay, so just to finish up for the lecture today, probably about 10 minutes or so. If we don't get to end replication, that's not a problem. We can do that on Thursday. Is just to look at some of the differences between prokaryotes and eukaryotes. Oh, and this is a nice little summary. I'm not going to go over that because you can look over that yourself. Do we have any notes in here? Oh, yeah, we do. Okay, so in eukaryotes, there are some nuggety differences, right, which are important. The general principles are the same, right? The whole DNA gyrase, helicase, needing to make an RNA primer, leading versus lagging strand, DNA ligase, sealing the nicks, things like that. Those general principles are the same. The differences come down to basically the difference in how eukaryote DNA is structured and packaged. And so the big one is that eukaryotic chromosomes are linear. They're a straight line, which does cause some issues later. And also, eukaryotic DNA is rarely found as just bare DNA like it is in prokaryotes. It's always complexed with proteins to make something called chromatin. And this is a definition that's worth posting up. Did I spell that right? No, I didn't spell that right. Crap. Yeah, so we need to deal with those issues if we're to replicate DNA, eukaryotic DNA. Also, there are some slight differences and wrinkles in how these things get done, right, which we'll talk about in a little bit. So first up, eukaryotic DNA, like human DNA essentially, is made up of, or is found as a complex with histone proteins. And there are typically two forms. One is this form, I always think of this as like a meatball, essentially, and it consists of 4 pairs of histones. So we have 8 proteins total, and you can kind of see that over here in this image. And then you have an extra one, which kind of basically kind of keeps the DNA wrapped around the histones and also spaces them out so they're able to kind of move around each other. And that's our histone H1. That's a linker histone. So if you want to replicate eukaryote DNA or human DNA, you have to kick these histones off, right? You have to be able to not just unwind DNA, but first access the DNA, which means typically removing some histones, and then you can unwind DNA and start the whole replication process. And then once replication is finished, you have to put all those proteins back again, right? And a whole bunch of extra ones, right? Because you've got twice as much DNA. So another difference is that, well, eukaryotes have a whole bunch of DNA polymerases, and I'm not going to go into all of them now because I just don't know all of them. But essentially, in prokaryotes, you have a primase and then DNA polymerase 3. So primase makes the primer, DNA polymerase 3 comes in and extends the DNA. In eukaryotes, you have an initiator DNA polymerase, right? That's DNA polymerase alpha that comes in. It makes the primer and a little bit of DNA polymerase, newly synthesized DNA, and then it falls off, right? So it just does the very beginning of DNA synthesis. And then when it falls off, a second DNA polymerase comes in, depending on whether it's lead-in or lag-in strand. Lead-in is epsilon, lag-in is, delta. And that continues that replication on for the bulk of the rest of the synthesis, right? So it's not it is 2 distinct enzymes, but in eukaryotes, both of those enzymes are DNA polymerases. In prokaryotes, it's an RNA polymerase called primase and a DNA polymerase. So that's just kind of one of the extra wrinkles. And then the final wrinkle, and I think we'll actually end up with just slide 23, because yeah, talking about telomeres and stuff is a big enough topic to want to spend time on it. So we'll do that on Thursday. And then another wrinkle is how that RNA fragment is removed, right? And how that process is finished, right? So same problem occurs. We need to get rid of that RNA, and we need to seal the backbone, right? Join those fragments together. So in prokaryotes, we have that DNA polymerase 3 falls off, DNA polymerase 1 comes in, and it does the primer removal and extension of that little section. In eukaryotes, humans, let's just call them humans, it'll be a lot easier. It doesn't quite work that way. Instead, what happens is the DNA polymerase, whether it's delta or epsilon, right, comes up and it hits the RNA primer. And so essentially it can't continue anymore because there's no free template DNA to copy. And so the RPA, what does that stand for again? Replication protein A, that's it. Comes in and it actually levers that primer off of the template strand, so that DNA polymerase can continue, and eventually that RNA primer is just removed entirely with a little bit of DNA. And so the DNA polymerase epsilon or delta basically continues along to the end. It's almost like instead of it being a new protein or polymerase that does it. It just kind of pauses, waits until the blockage is out of the way, and then just continues what it was doing originally. So that's kind of the big difference between eukaryotes and prokaryotes. And then once that space is synthesized, then we have DNA ligase come in and it seals the backbone as it does in prokaryotes. And I think that's more than enough for today, to be honest. My ears are achy. I'm sure your brains are tired. So I'm going to call it a day for today, and we're going to meet back at 2 p.m. for lab. So what I'm going to do for the first part of lab, perhaps, I don't know, 15, 20 minutes, something like that. Depends on need and who shows up. We can go over the standard curve assignment that's due tonight. So if you've already done that, and I think some of you have had it graded, I'm not sure, not very many, but some of you have talked to me about it. Have a slightly longer lunch. And then we'll talk about the Excel stuff for Thursday. So remember, again, if you want help with the standard curve assignment that's due tonight, meet me back at 2, and we'll go through that. And then, oh, cool, that's fine. And then we'll go over any questions that you may have for making that nice graph for the Excel assignment for Thursday. All right, I'm going to go grab myself some lunch and see you all in a little bit.